Hello, welcome to Module 6 of third week of our course. In this course, in this module, we will discuss about writing ODE based equation for a very common process in cell biology, that is transcription and translation, which ultimately controls the concentration of proteins inside a cell. So if I look into the steps, simplified steps involved in transcription and translation, I can write a scheme that's shown here in the slide. I have a gene G, which is under the control of a promoter. A transcription factor or an external signal comes, activate this promoter, and a mRNA M is formed. Remember, mRNA is formed by transcription, shown by this arrow. Now, as once a mRNA is formed, that can get degraded by this method. So I have degradation here, and by translation, mRNA is getting is giving rise to protein P, and protein P can also get degraded. So this is a generalized scheme, very simplified. You can have much more complications inside the whole thing. For example, in eukaryote thing, your promoter may be condensed, it may have to open, and all these issues are there. And for example, in case of translation, you may assume that ribosome comes, binds, scan the initiation site. All these things we are not considering. What we have done, we have clubbed them together, and we have created a simplified schematic representation here, and we will try to write the ODEs based on this. Now, if I don't require to separate out transcription and translation as a separate entity, in some cases I don't need, I just am interested only in the dynamics of the protein, and I'm not much bothered about how the whole process is broken down in different parts, I can write a simple ODE representing the formation, the rate of change of protein concentration, and we have seen it earlier that it will be equal to Ks into S, that is the rate constant of synthesis of the protein, into S, that is the signal which is inducing the production of the protein, minus Kd into P, this is the first order degradation of protein. You have seen it in an earlier module. This is the simplest one I will always recommend or try to use if I am not bothered about the detailed mechanism, multiple steps involved in the production of a protein. But in some cases, actually I have to bother about these multiple steps in transcription and translation separately, because maybe I want to explore the dynamics in terms of the dynamics of the external signal. So remember, this external signal or the transcription factor which goes and binds to the promoter and controls or induces or inhibits the expression of the transcription of a gene may have a particular dynamics, and I want to know the input-output relation in this transcription induction or inhibition. So in that case, I cannot club transcription and translation together. I have to separate them out. So I have to consider transcription as a separate process in which the input signal S or the controlling transcription factor will have a role to play, and translation as a separate process. Some cases, and many a time actually we forget that actually the degradation or stability of mRNA is very crucial in the dynamics of the protein as a whole. So suppose I want to study the effect of stability of mRNA on the dynamics of the protein in concentration in the cell, then I have to consider transcription and translation separate, because I want mRNA as a dependent variable also. So similarly, if I want to look into the rate of translation as a separate feature parameter, and I want to study the effect of different rate of translation on the dynamics of the protein, then I may have to separate out transcription and translation separately, and I have to model them and write the ODE separately. So if I separate out transcription and translation, then I will consider mRNA and protein as separate dependent variable. So I will write one ODE for mRNA, one ODE for the protein. So let us try to do that. I will start with writing the ODE for the mRNA. Now, mRNA concentration is controlled by transcription and degradation of the mRNA. Now, transcription is again controlled by the signal, which may be itself a transcription factor or a molecule which goes and activates a transcription factor. So that transcription factor or inducing signal will control the rate of synthesis of the mRNA. For an inhibitory signal, it will also control the rate of synthesis of the mRNA. So that means I have to incorporate this signal into the ODE for the mRNA. And for example, I have written here, it is a very common thing. You add IPTG in your bacterial culture where the bacteria is carrying a plasmid having a lac operon and controlling the expression of a gene. So you vary the concentration of IPTG and the amount of the gene transcribed changes. So I can write a simple ODE the way I have shown here. dm/dt is equal to, that is, M is the concentration of the mRNA, M is a concentration of mRNA. So the change in rate of change of concentration of mRNA is equal to K1 into S. K1 is the rate constant for transcription. S is the signal. For example, concentration of IPTG added to the culture. Minus K2 into M. K2 is the rate constant for degradation. M is the concentration of free mRNA. So this is the first order degradation. So the first part is synthesis by transcription. Second part is degradation. Now see the first part, the transcription part, K1 into S. So that means if I keep on increasing S linearly, then the rate will also increase linearly. But the fact is, in many experimental systems, it has been seen that this relation of rate of transcription with the amount of input signal is actually not linear. Rather, it is non-linear and particularly sigmoidal, S-shaped. As I have shown here in these graphs, in many experiments, for example, in experiment involving IPTG as an external signal for inducing the expression, when inducing signal is increased, the rate of transcription does not increase linearly, rather follow a sigmoidal path as I have shown and eventually saturates. It does not increase further. If you increase signal further, it will still remain here at the saturation. So it is a non-linear behavior. Similar has been observed for many transcription factor molecule factor which goes and inhibit transcription of a particular gene, where inhibitory signal, if you increase, the rate of transcription does not decrease linearly, rather non-linearly in a S-shaped sigmoidal fashion, and it is just reverse of the previous one. So that means I have a non-linear relation between signal S and the rate of transcription. And it is now well understood that in many cases this nonlinear relation, this sigmoidal behavior arises because transcription factor works with cooperativity. One transcription factor does not work alone, it binds with multiple copy of the same transcription factor, then make a large complex, and that complex is functional. Many a time, not just one molecule is involved, one transcription factor goes, attached to the promoter, and then it recruits other transcription factor to create a large complex. Unless until this large complex is formed, the effect of the transcription factor is not manifested. So it is a cooperative phenomena. And as I have a cooperative phenomena, I have this sigmoidal behavior. Now, that means if I have a system where transcription rate have a sigmoidal relation with input signal, I cannot use this simple linear relationship of K1 into S. So how can I modify this equation to reflect the sigmoidal behavior that people are observing in experiments? Now remember, different sigmoidal function you can write mathematically, large number of them. You can make an imagination and write a function which will have sigmoidal input-output behavior. But we have to choose a function which will be sigmoidal in behavior, at the same time should be very simple and has some biological meaning. And in case of modeling biological system, we very frequently use Hill function to represent a sigmoid function because it is very simple and it has less number of parameters, and those parameters can be actually determined from experiments. So let us look into what is Hill function. Hill function is nothing, y, that is maybe the output, is equal to x to the power n divided by k to the power n, k is again a parameter, and plus x to the power n, n and k are parameters. Means these are constants. And I have to get those values, either I have to guess them or from experimental data I have to get them. If you look into this function, you can easily see if x varies from 0 to any higher value, positive value, y will increase initially when x equal to 0, obviously y equal to 0, but as you keep on increasing x, y increases, but eventually when x becomes very large with respect to k, this y becomes nothing but equivalent to x to the power n by x to the power n, because x has now become very large with respect to k, so this becomes 1. So that means this function y changes from 0 to 1. 1 is the maximum value. And that helps because it is within a particular range. And if you look into the diagram here shown in your slide, I have varied x in a positive value from 0 to 300 and y in the vertical axis. You can easily see this blue curve is a sigmoidal one. Here I have considered n equal to 3 and k equal to 25. So now look into this parameter k and n, what they are. Before I move into, I'll just remind you that you may have seen this type of Hill function in biochemistry textbooks where hemoglobin has been discussed. Hemoglobin has multiple subunits and they have cooperativity, and the partial pressure, the oxygen saturation of hemoglobin has sigmoidal behavior, and Hill function is used there to represent that sigmoidal behavior. You can look into that. Now let us go back to the parameters k and a. K is called Hill constant and it represents the position of half saturation. Let us look into that. I will change the value of k from 25 to something else, say suppose 100. Then I get this pink curve. This gray line is representing 50%, 0.5. So it is intersecting this blue as this value, which is nothing but 25. And this gray line is intersecting the pink line at 100. So that is value of k. So 0.5 is half saturation because the maximum value is 1. So half saturation is 0.5. So and k represent where I reach that half saturation value. And you can easily imagine that by experiment I can actually measure this half saturation value. That means I can measure the amount or the value of x for which the value of y will be 0.5, the half saturation, and that is my k. And this k is called Hill constant. N is interesting parameter. N is called Hill coefficient. And it decides how stiff this S-type figure would be, how stiff this sigmoid curve would be. If you take n equal to 1, I don't get much stiffness. I get a rectangular hyperbola type thing. Let us take the value of n. What I have done here, remember k is same in both the curves. That is 25. So half saturation is at 25. But I have changed in blue n is 3 and in the green one n is 1. So I have reduced the value and taken the value of n, the Hill coefficient, to 1, and you can see the curve has lost its sigmoid behavior and it has become a rectangular hyperbola. And in fact, it will be like that till you have n equal to 1, and if you have n greater than 1, then I start getting this sigmoidal behavior. So Hill coefficient tells me whether the system has sigmoidal nature or not, and if it is sigmoidal, how stiff that sigmoid is. Whereas k tells me that how sharp this sigmoid rise. Is it rising very fast with respect to the value of x? Because it determines the half saturation value at which we reach the half of the value of y. So using these two parameters, k and n, I can actually manipulate the Hill function and get different sigmoidal behavior, and that helps me in my modeling. So I have a simple function y equal to x to the power n divided by k to the power n plus x to the power n, and just two parameters. I can measure this parameter from experimental data and it gives this function, depending upon the value of n and k, give me large number of sigmoidal behavior. Now remember, for induction it is sigmoidal rise. As inducing signal increases, you have sigmoidal rise in the output, the rate of transcription. But for an inhibitory signal, the rate of transcription drops. So I require another function. I can actually tweak the Hill function and get this inverse Hill function where you don't have x to the power n on the numerator. You don't have it. So rather you have k to the power n, that is a Hill constant to the power n, divided by the same numerator, k to the power n plus kx to the power n. Again here, when x equal to 0, y will be 1 because that's what you want when there is no x maximum value, and then as you increase x from 0, y start dropping and it reaches towards 0. So this inverse or negative hill function I can use when I will have an inhibitory signal or a transcription factor I have in the system which goes and inhibit the process. So once I have these hill functions, inverse and the normal hill function in hand, let us try to model a transcription translation unit using this hill function. So what I have the same scheme. S is the signal, this I can consider as an input, which gives a positive induction for transcription of the gene G, and I get the mRNA M, which get degraded and which may get translated by ribosome forming protein P, and this is my output. So both mRNA and protein can get degraded, so they are getting degraded here. And I want to write ODE for both M and for P because I am considering them as separate entities, separate dependent variable. So now I will use hill function. So dm dt, rate of change in concentration of M, is equal to K2 into the hill function here, where S is the signal, into K to the power n plus S to the power n. Look into this part very carefully. When S equal to 0, K2 into this S to the power n by K to the power n plus S to the power n will become 0. When S is greater than 0, then this function will have this behavior, sigmoidal behavior, depending upon S. So this function has then. So that means as S will increase, this whole term will also increase and reach a maximum value K2. Because the hill function has a maximum value 1, so 1 into K2 is K2. So initially when S equal to 0, K2 into 0, so rate is 0, no transcription. As you increase S, S to the power n divided by K to the power n plus S to the power n, this hill function will move towards 1 depending on the value of S in a sigmoidal fashion. So when I am multiplying that with K2, the overall rate of transcription is also behaving sigmoidally and reaching the maximum value K2 at the maximum value of F, that is the hill function. So this rate of transcription has a sigmoidal behavior now with respect to S, and K3 into M is nothing but first order degradation of the mRNA. Let us look into the protein part. So dp dt is the rate of change in concentration of protein will be equal to now K4 into M. M is the amount or concentration of mRNA present. So obviously translation will depend upon how many copies of the mRNA is present at that time for that particular protein. So it is K4 into M, first order reaction, minus K3 in K5 into P, that is the first order degradation of the protein. So I have a simple two ODs. The first thing only has this hill function and the transcription rate control. Now we have to remember one critical issue here. Let us clean this part a bit so that you can see it clearly. If I consider the rate of transcription is equal to nothing but K2 into S to the power n by K to the power n plus S to the power n, the way I have done here, then when S equal to 0, rate is also 0. That means when there is no signal, there is no production of the mRNA for this gene. So if there is no mRNA, there will be no protein production. But that may not be true in many cases. In fact, for a IPTG based system also, even in absence of IPTG, there is some amount of transcription from lac operon, and that transcription and translation of the protein is actually required to run the lac operon system. So in some times, even in absence of the inducing signal, there is low but steady expression of the mRNA. We call it leaky expression. And sometimes for the physiology or for the dynamics of the whole system, actually that leaky expression is required. So how can I represent it? Because the ODE that I have written here does not incorporate that. Because when S equal to 0, rate of transcription will be 0, there will be no formation of the mRNA, so there will be no formation of the protein also. I can simply change this equation by adding a constant term, K1. K1 is the rate for leaky expression. So, when S will be 0, then this Hill function will become 0. So, K2 into Hill function will be 0, but K1, which may have a very low value, it will give me rise of mRNA, it will keep on producing mRNA. Usually, we take K1 as around 0.1 or 0.01 of K2. So, 10% of or 0.1% of the maximum rate of transcription that can happen by induction. You take a very small value of K2 with respect to K1 with respect to K2. So, I can simply change this ODE to this one and incorporate K1. So, my rate of change of M, that is mRNA, dm dt becomes K1 plus K2 into the Hill function involving S minus K3 into M. So, if I have these two now, the first one considering leaky expression and the second ODE is for my protein. So, if I have this, I have two ODEs. Now, imagine I have a cascade of transcriptional network, means one protein is produced and then that protein acts as a transcription factor for the next protein and induces the production of that protein. That protein also works as a transcription factor and control expression of another protein. So, if I have multiple transcription translation unit in my model, then for each case I have to write these two ODE, one for mRNA, another for the protein, and that may complicate my calculations. So, I will try to reduce my ODE. So, how can I reduce my number of ODE for a transcriptional translation system? That is what I will discuss now. We all know that usually transcription is a faster process than translation, particularly in case of eukaryotic system, because transcription happens inside the nucleus and then the nucleus has to get it transported outside the nucleus into the cytoplasm where the ribosome will bind and many other processing of mRNA will happen before that. So, if I do not consider nucleus and cytoplasm as a separate chamber, separate compartment, and as a whole I consider cell as a single unit, then I can consider that the production of proteins is slower with respect to transcription. So, I can safely consider that before the protein production reaches a steady state, mRNA production reaches a steady state, because mRNA is produced in a faster rate, it also gets degraded, so I reach a steady state. So, what I can do? I can assume that if transcription is much faster than translation, mRNA will reach steady state very fast. That means I can consider this dm dt is equal to 0. So, let us do that. So, I can consider steady state for the mRNA. So, I can put dm dt is equal to 0. That means this whole relation K1 plus K2 into the Hill function minus K3 into M is equal to 0. So, this M is the steady state concentration of my mRNA. So, I want to separate that out from this algebraic relation. So, I take K3 and M in left hand side and keep everything else on the right hand side. Further, I can divide both sides by K3, then I get the steady state concentration of M. This is my steady state concentration, M is equal to K1 by K3, considering that I have a leaky expression, plus K2 by K3 into the Hill function, S to the power n divided by K to the power n plus S to the power n. This is my Hill function. So, now I can rewrite the whole thing, because I have reduced one ODE. I do not require a ODE for the mRNA, because I have considered that it has reached a steady state. I am left with only the ODE of protein. So, dp dt, and I will place this value of m here, replace this value of m with the steady state value of m. That is what I have calculated just now using this relation. So, I will replace that. So I can write from this relation, I can write dp dt is equal to K4, there is a rate constant for translation, into this is the steady state concentration of at steady state that just we have calculated, K1 by K3 plus K2 by K3 into the Hill function minus K5 into P. Now I will club multiple parameters together. I will take K4, K1 and K3 together, K4, K2, K3 together. I can replace this one by a new parameter, I can replace this one by a new parameter, and I get this relation dp dt equal to Kb, that is the basal rate of production. This is my basal rate of production plus Ki, there is the rate constant for inducible production of the protein, Ki, into the Hill function involving the signal S, that is S to the power n divided by K to the power n, K is the Hill constant, plus S to the power n minus K5P, that is the first order degradation. I have written down, spelled out here, Kb equal to K4 into K1 divided by K3, whereas Ki is K4 into K2 by K3. So I have only one ODE now. So this ODE will give me the dynamics of the protein when the input signal is S and the input output relation at the transcription level has a sigmoidal behavior. Now suppose the signal inhibits my production, inhibits my transcription, then I have to use the inverse or negative Hill function, and I can write the ODE the way I have shown here. dm dt equal to K1, that is the leaky expression, rate of leaky expression, plus K2 into the inverse Hill function here, or negative Hill function here, minus K3 into M. K3 into M is nothing but your rate of first order degradation of mRNA. And the second ODE that is shown here, dp dt, is K4 into M, that is the translation of the rate of translation of the protein, and it depends upon the concentration of mRNA present, minus degradation of the protein, K5 into P. Now just like the previous one, here also I can consider the transcription is much faster than translation, and the transcript, the mRNA reaches a steady state much before the protein reaches a steady state. So I can consider this dm dt is equal to 0, dm dt is equal to 0, and I can replace m steady state value with some algebraic relation from this first ODE and put that one in the second one, and I can get, just I have done the way I have got in the last slide, dp dt, there is a rate of change in protein concentration equal to Kb, this is the basal rate of production, plus Ki into the Hill function, inverse Hill function or negative Hill function in this case, minus K5 into P. So if I jot down what we have discussed in this module, we have discussed that the protein production and degradation, usually we may consider transcription, may not consider transcription and translation separately, I may club down together in a single process, but sometime we have to separate them, consider them as a separate process, and in those cases I have to consider mRNA and protein as separate dependent variable. That means I will have two different ODEs for them, and for this I have considered transcription and translation as separate process. Now if I do that, it allows me to investigate role of different inducing and inhibitory signal in transcriptional process. It also helps me to understand the effect of stability of mRNA on the level of protein. All these questions can be approached. Now we have to remember, in most of the cases, transcription factors or inhibitors work with cooperativity. So if you have cooperativity, this cooperativity sometimes leads to a sigmoidal input-output relation. So input-output is not linear here, but non-linear and has a S shape, and the S saturate as a higher value. If you have that type of behavior, if experimental data is telling you that you have a sigmoidal input-output relation for the transcription factor involved in this system, you can use Hill function, which is very simple, or an inverse Hill function to model this type of sigmoidal behavior. And remember, Hill function is defined by two parameters. One is called Hill constant, which represent the half saturation value, where I get the half saturation, and the Hill coefficient n, which tells me whether I have the sigmoidness or the S shape in the curve or the sharpness of rise of the curve. If n is equal to 1, I get a rectangular hyperbola. I don't have a sigmoidal behavior. That means I don't have any cooperativity in the system. When you have cooperativity in the system, when the transcription factors works with cooperativity, you will have sigmoidal nature and n will be greater than 1. That's all for this module. Thank you for watching.